Have you ever imagined that in a simple university laboratory, scientists could create a new form of life that never existed before on Earth? It sounds like science fiction, but in 2008, a group of researchers achieved something extraordinary. They witnessed the birth of a new species in real time, challenging everything we knew about evolution and the origin of species. This is a story about how science sometimes pushes the boundaries of our imagination, proving that reality can be more fascinating than any fiction. Since Charles Darwin published On the Origin of Species in 1859, scientists believed that the emergence of new species was a process that took thousands, if not millions of years. It was like trying to watch a tree grow. You know it's happening, but it's impossible to see the process in real time. At least that's what we thought. In a University of Michigan laboratory, a team led by Dr. Chelsea Spencer was about to challenge this fundamental notion. What started as a routine experiment with yeast, those same microscopic organisms we use to make bread and beer, would become one of the most revolutionary discoveries in modern biology. The experiment began with a simple question: how long does it really take for one species to split into two? The team decided to work with yeast for several practical reasons. They reproduce quickly, are easy to manipulate in the laboratory, and most importantly, can be preserved by freezing, allowing scientists to go back in time to study previous generations. The researchers established specific conditions in their laboratory, replicating an environment that would force the yeast to either adapt or perish. They created a culture medium with limited resources, where different yeast strains would have to compete for nutrients. What happened next surprised even the most experienced scientists. In just 500 generations, a blink of an eye in evolutionary terms, a new lineage of yeast emerged. This new strain not only adapted to the challenging environment, but also developed unique characteristics that made it genetically incompatible with its ancestors. In other words, a new species had been born. The most fascinating aspects of this experiment were the unexpected discoveries that emerged along the way. The new species developed the ability to metabolize resources that its ancestors couldn't utilize, effectively creating its own ecological niche within the laboratory. It's as if we had witnessed the microbial equivalent of the emergence of the first mammals among dinosaurs. The researchers discovered that the new species had developed a completely new chemical communication system, something never before observed in yeast. It was as if they had invented their own language. Even more surprisingly, when scientists tried to make the new species reproduce with its ancestors, they discovered that changes in just 14 genes were responsible for reproductive incompatibility. To put this in perspective, it's as if a small change in a few lines of code could create an entirely new computer program. The implications of this discovery are truly revolutionary. Imagine a future where we can create specific organisms to clean polluted oceans, developing them in the laboratory to consume plastic and other contaminants without affecting existing marine life. Develop new yeast strains capable of producing complex medications more efficiently and economically, making treatments more accessible to millions of people. Better understand how species adapt to climate change, allowing us to predict and possibly mitigate the impacts of global warming on biodiversity. This experiment has opened the doors to a new era in evolutionary biology, where we can not only study evolution, but also direct it in ways that benefit humanity and the planet. Think for a moment. We're living in an age where we can witness and even influence the process that Darwin spent a lifetime trying to understand. It's as if we've gained a front row seat to the theater of evolution, where the show doesn't take millions of years, but happens before our eyes. And the implications go far beyond the laboratory. This experiment makes us question, if we can create a new species in such a short time, how many species are naturally emerging around us without us noticing? Could it be that, while you're watching this video, a new species is being born somewhere on the planet? Even more intriguing, If evolution can happen so quickly under controlled conditions, how can we use this knowledge to help endangered species adapt to climate change? Or to develop organisms that can help us face the environmental challenges of the 21st century? Scientists around the world are just beginning to explore the possibilities opened by this discovery. Some laboratories are already applying similar techniques to study the evolution of antibiotic-resistant bacteria, seeking to develop more effective strategies to combat superbugs. Investigating how different environmental pressures can accelerate or slow down the speciation process, providing valuable insights into how biodiversity develops in nature. Developing new methods to predict and potentially direct the evolution of beneficial organisms for agriculture and medicine. This discovery isn't just a scientific curiosity, it has profound implications for our understanding of life and our ability to face global challenges. In agriculture, we can develop crops more resistant to pests and climate change in a fraction of the time it would take through traditional breeding. In medicine, there's the possibility of creating microorganisms specialized in producing medications or fighting specific diseases. In environmental conservation, we can better understand how to help endangered species quickly adapt to changes in their habitats. Of course, with great power comes great responsibility. This scientific advancement also raises important questions. How do we ensure that laboratory-created organisms don't cause ecological imbalances if released into nature? What are the ethical limits for creating new forms of life? How do we balance the potential benefit to humanity with potential risks? What do you think about this revolutionary discovery? How do you imagine the ability to create new species in the laboratory might change our future? Leave your opinion in the comments below, and don't forget to subscribe to the channel for more fascinating content about science and discoveries that are changing our understanding of the world. Share this video with your friends who love science, and let's explore the frontiers of human knowledge together. Hit the bell icon to never miss any updates about the most impressive discoveries in modern science. To access the sources cited in this video and additional material about the experiment, check out the links in the description. See you in the next video, where we'll continue unraveling the most fascinating mysteries of science.